So first off, I want to thank Dr. Baumel and Hosborgai for inviting me to give the talk, and obviously to Jack West and the entire Grace group. And it's really a pleasure to be here with everybody. My name is Josh Sabari. I'm a medical oncologist at NYU, and it's great to see familiar faces, people who I've taken care of and who've taken care of me also in the past. I was telling people in the audience that they didn't know how nervous I was meeting you too. So. And it's a pleasure to be here to talk to you guys. So what I'll be talking about is sort of a broad overview and really challenges and opportunities in clinical research with narrow populations. And I'm going to try to avoid going into any major detail on any specific, or I don't want to bore you guys with the sort of studies from or the slides with curves and survival, but I really want to talk about general or major concepts. So first off, I have a few disclosures. I'm consulting advisor from AstraZeneca, travel funding from Merck, and Resolution Bioscience, and research funding from Merck. So you've all talked about this and all thought about this. So history of targeted therapy and the driver discovery and how far we've come over the last 15, 20 plus years. So in 1999, we really knew about KRAS mutations, and 75% of our patients were unknown. Right? And everybody was getting chemotherapy, and we'll talk a lot about that. And then moving towards 2004, we identified EGFR mutations, and Dr. Yu, Helena Yu, will talk about that story. And then, in the last 3 to 5 years, or maybe a little more than that, 5 to 7 years or so, the landscape has sort of expanded. So the common alterations that have FDA-approved indications, we all talk about as EGFR, as ALK, as ROS1, and BRAF, but there are so many other alterations that we are now identifying in patients and are able to match them to targeted therapies, either on a clinical trial or drugs that are NCCN or National Cancer Center Network recommended medications. And I know a lot of times patients have a hard time getting drugs covered and funded, and we can talk a little about that and have a discussion. So this is a paper from 2018, actually, by Emmett Jordan, who was a fellow at the time at Memorial Sloan Kettering, looking at their MSK impact data set. And you can see how diverse or heterogeneous the patient population is. A lot of folks ask about EGFR here. Why is it so high? Why is it 28%? And the answer is because of Helena Yu. That's the answer, because she's there. No, because this is a sort of selected patient population. We're sequencing patients and looking for abnormalities. So not everybody was being sequenced originally in the initial series. But you can see, 1.7% RET fusion, MET splice site, 3%. Other alterations like HER2, for example, 2.3%. So we know that the pie is actually a lot bigger. We're split into a lot smaller slices than we had initially thought. And really the unknown is a lot less now. So the first point I'll make is it's critical for anybody newly diagnosed. It doesn't matter whether they're a smoker or a non-smoker. Yes, the rates of these alterations are higher in non-smokers, but they should always have next generation sequencing done. It's controversial if it should be done on tissue or plasma. I do recommend plasma actually and tissue in all my patients, and I'm sure that's been brought up or has been brought up or will be brought up if not brought up yet. So where were we 5 years ago when somebody was diagnosed with a stage 4 lung cancer? All we had available was chemotherapy. Okay? You could argue that we had EGFR inhibitors, but really it was chemotherapy in the frontline setting, and if there was progression or the cancer grew, in the second line setting we had chemotherapy as well. And I spoke to somebody earlier today, I don't know if she's still in the audience, she said somebody offered her docetaxel in the second line setting, and she said it would work about 7 to 10% of the time. And what did you do? So I mean, we've come a long way over the last, 5 or 6 years. So this is sort of a paradigm now. I won't go over it in any detail, but chemotherapy is available, immunotherapy is available, and a lot of different targeted therapies are available. And this is a slide compliments to Dr. Greg Riley. It's somewhat updated now, and Ipi-Nivo is somewhat controversial, as Josh and I had talked earlier in our session. This is an interesting figure from a recently published review in Nature Review Clinical Oncology, just looking at the drugs approved in the last year in oncology. And I've highlighted here in the box the drugs approved in lung cancer. So these are indications in lung cancer that were not approved or newly approved. And it's fascinating to me, as even as a fellow, we would see maybe one drug approved a year. We'll talk a little about next-generation sequencing to identify driver alterations, and I explained that that's critical for all folks with newly diagnosed lung cancer to obtain, to understand the biology or the genetics of their cancer. And then obviously we mentioned cost both to an individual but also to society. And then how do you prove benefit of a drug, right? Especially if it's a very narrow patient population, maybe you're missing most of the patients that have the abnormality. How do you prove benefit? Is it on a clinical trial where you're looking at safety, tolerability? Is it the response rate of the drug? Is it how long people have, a disease-free interval or do not progress? Or is it actually looking at real-world outcomes, right? Actually, people getting the drug in the community, not on a clinical trial or off-label. Is that something important to look at? And I think actually we are moving more and more towards real-world outcomes in rare alterations and rare, mutation space, because we're not able to do clinical trials effectively at single institutions or even multiple institutions. So we're now starting to pool data, both prospectively but also retrospectively. And I think, interestingly, in the next 5 to 10 years, we will see more of our clinical trials actually being done in an EMR-type setting with using control arms, but also looking at patients, how they're doing prospectively, especially patients who are being treated with medications off-label or in expanded access or compassionate care use. So when we think about challenges, one of the major questions to bring up is who are the stakeholders here? So obviously one, always is the patients. And I've met, a few of the groups here today. It's so critical that you guys speak up, right? And that you guys drive the science and that you guys are involved at the table. So one of the first things I did as a fellow, or was fortunate to do as a fellow, was I attended an ACR ASCO clinical cancer research, methods in clinical cancer research, where I was taught how to develop and write a clinical trial. And some of you folks in the room know Roy Herbst was in the room, Jyoti Patel was in the room, and then I won't mention names because I don't know if they want to be public, but there were 2 patient advocates in the room, and they were the toughest folks in the room. They ripped my clinical trial to shreds. But it was the first time. I won't mention any names. It was the first time that I realized, right, how important that was to actually have the patient at the table when we're thinking about the design of trials and we're thinking about sort of how this is going to affect quality of life, care. Is this something even feasible? Can we biopsy somebody 3 times? Is that an okay thing to ask of somebody, right? And I'm not going to weigh in, but you have to be there at the table when we're making those decisions. And also family and caregivers. So I run a lot of phase 1 studies now at NYU, and obviously you guys know here in the room, without family members, without caregivers, there's no way that people can get care and no way that patients can be able to come in and tolerate side effects, for example, or just be able to emotionally deal with everything going on related to their disease and their family life and their home life. Research sponsors. So this is actually really controversial, right? And anyone reading the news knows that this has been a sort of touch and go sort of thing. So how important is industry? We all know it's very important, right? They're developing the drugs. I think we need to partner with them, but we need to be more on an even playing field. Academia, obviously, government, nonprofit organizations, and again, the patient advocacy groups I think are critical, and we'll talk a lot about the EGFR and ALK space briefly about what's happening in that space. So, and also how can we work together? I think hearing from folks and hearing about your ideas always enhances our practice and always enhances individual patient care for yourself and those folks who come after. So this is sort of the paradigm in 2019 for how we develop drugs. And it's actually, the point here is that most potential new medicines do not end up ever treating large numbers of patients, for many different reasons. In the basic arena or the drug discovery area, the preclinical, a lot of the clinical assets fail, meaning they don't show activity or they show some toxicity in the early setting. And then once drugs enter phase 1, so just a quick raise of hand, has anyone been on a phase 1 trial in the room? Thank you. So can I mention the phase 1 that you were on, because you had mentioned it to me?
Yeah, lorlatinib, phase 1.
So because of your participation in that phase 1 study, lorlatinib will likely be FDA approved, right? I mean, so I apologize. There you go. So I apologize. But because of you, you allow folks to come after who will benefit from this drug. So then in the phase 2 setting, you're looking at efficacy, and then in the phase 3 setting, you're looking at comparing versus the standard of care, standard control. And then again, moving back to the rare alterations, things like RET and things like MET exon 14, are we powered? Can we do phase 3 studies, right, where we're able to compare groups of patients who get a drug versus patients who don't get a drug, placebo, versus standard of care or chemotherapy? And is that even fair to do when we have such a good biomarker or such a high pretest probability that patients will respond to specific drugs? So I think the idea of the FDA changing their mindset of how drugs can now be approved, instead of going through this whole large process, we're seeing more and more drugs being approved based on very, very robust phase 1 data, accelerated approval, or open access programs for that matter. So here's actually the biggest problem, right? So these are folks with lung cancer, right, and on the screen here. And we talked about EGFR, and we talked about ALK, and KRAS is a common alteration that has been very difficult to target. But when you look at the more rare alterations, like RET, BRAF, or ROS, for example, we're dealing with very small numbers of people. Okay? And it's very, very hard to design and develop specific clinical trials for this subset of patients. And I think what we need to do is we need to sort of, like we mentioned, more robust access on phase 1s, not only at academic centers. So you had mentioned you were being followed by somebody phenomenal at Hopkins, right? But this drug was unfortunately not available there. So I think what we need to do is we really need to partner with our pharmaceutical partners in saying, how can we get these drugs to more patients? Maybe not on a robust, phase 1 group. Maybe once we have a signal, we can do expansion or early access for patients, and the data might not be as clean or it might not be as robust, but it might be able to help patients with their diseases. So going back to the curve where we are now, again, so much more diverse than where we were back in 1999 or even 2004 for that matter. I want to briefly talk about TRAC because it is one of the most rare fusions, and I think it's one of the success stories in my mind. Other people would argue that it's not a failure on the system, but a sort of concept of how a subset of patients with such a rare alteration can have a drug approved in such a rapid fashion. So TRAC fusions are actually found in diverse cancer histologies. So you can see in brain cancer and thyroid cancer, but really most commonly in salivary secretory breast cancer, and interestingly in pediatric patients, infantile fibrosarcoma and congenital nephroma. But we also do see these occur in lung cancer. And the question is, how frequently? And I don't think we have a good sense of this. And we'll talk briefly about some estimates. But in total, the estimate of all patients in the United States are diagnosed with a TRAC fusion or who have TRAC fusion is about 1,500 to 5,000 patients a year. And that's, the reason for that range is perhaps we're not identifying all patients. Not all patients are receiving next generation sequencing at this time. This is just from an organization looking at what the estimated frequency is. Actually, the maximum numbers in orange or yellow, I'm a bit colorblind, I apologize, and in purple, the minimum numbers. And if you go down to lung cancer, the max is about 3%, and I think we can all agree that that's relatively high. I've only seen one patient in my clinical practice with an NTRK fusion. And then the low or the minimum estimate is about 0.2%. So we're talking about less than 300 to 400 patients a year in the United States. And it was a heroic effort to get this phase 1 study through. So this is a phase 1 study of this drug, larotrectinib, in TRAC fusion positive patients. And just to orient everybody, this is a waterfall plot. Each individual line is a patient, and when the line goes up, it means the drug doesn't work. And when the line goes down, it means the drug worked, meaning it was shrinking the cancer. And here on this side, you can see the level at which the cancer shrunk. And minus 100% is what we call a complete response, which is amazing. And this line here is what we would call a response on a clinical trial at minus 30%. So you could see most patients responded to the drug. So if you had that specific alteration, this drug was a phenomenal drug for that patient. So 55 patients enrolled, including both adults and children across multiple different tumor types, right? So you see thyroid cancer, colon cancer, melanoma, et cetera. So this is a critical concept in how we need to think about trials in the future. And based on this study, the FDA actually earlier last year approved lorlatinib for solid tumors with NTRK gene fusions, and this was only the second approval, pan-cancer, pan-indication. Usually most approvals, like I showed you guys earlier on one of those slides, are individual approvals in individual diseases. But again, given how rare these alterations are now, I think we need to sort of open our mind and think about this being a new norm and a new paradigm. So what are some of the opportunities that we have to do this work, right? So we need to partner with stakeholders, and we'll talk a little about partnering with our patients, family members, and specifically advocacy groups, some academic collaboration. So one of the things that I've been doing at NYU now is reaching out to all of my counterparts all across the country to pool our data, and this is something I learned from Alexander Drillon, Dr. Drillon. It's critical when you have rare alterations. You know, if you can have 4 or 5 of those in your whole career, that's not going to change or move the needle forward. But if you combine that with all of the oncologists in the US, for example, or in the world, you then can increase your numbers and actually benefit patients. And then also industry collaborations, I think, are critical, right? Because at the end of the day, they're driving a lot of the basic research, and they're also driving a lot of the drug development. Also, this changing paradigm of drug development we talked about. And lastly, we'll talk briefly about understanding mechanism of resistance, and we have Ivy Elkins here and Jill Feldman who really are critical in this space. You know, you guys are one of the first groups that I've seen where you partnered with an academic center to actually enroll patients on a trial, donating tissue to then better understand resistance mechanisms. And to me, I mean, that's how you're going to move this field. We're all, you know, all of oncology forward. So that's really impressive on your guys' end. So some of the groups, and I learned that not only is there ALK positive, but there's also ALK fusion, which is a group as well. And I apologize if I left everybody off, but these are some of the groups that I'm aware of and have interacted with patients and referred patients to. And thank you guys for the work that you do. So I wanted to end briefly just with one of my patients who I'm taking care of now to show you how important understanding the genetics are and how important sort of testing is and where some of the breakdowns happen in this process for individual people, right? These are people that we're taking care of in clinic. So this is a 46-year-old woman. She's a mom of 3. She's a bank teller, lives in New York City, never smoked. She came in with a fluid that was building up in her lungs that had come back multiple times. The first time they drained it, and the docs told her it was related to an infection. A month later, the fluid came back, and she was more short of breath, and they drained it again, but this time they looked at it under the microscope, and it revealed, unfortunately, a lung cancer or a lung adenocarcinoma, which is one of the most common types of lung cancer. And they did the initial testing for EGFR and ALK, and that was negative. And they also did PD-L1 expression, that was 90%. And the patient came to me with the recommendation of starting, interestingly, chemotherapy. And so I apologize. This is a patient that I had met after she started therapy. So when her initial blood plasma assay from next generation sequencing back in July of 2016 identified a ROS1 fusion, which everybody in the room is aware of, and we also confirmed that on her tissue, next generation sequencing assay, and the partner was the CD74 fusion. And again, this is a rare alteration that we see about 1 to 2% of the time. So this is a study from Alice Shaw looking at crizotinib use in this patient population, and the average or the median progression-free survival was about 19.2 months, which is significant, right? Compare that to chemotherapy is a lot lower. So this patient was started on crizotinib back in August of 2016, and she was being taken care of my colleague at the time, Dr. Lena Gandhi, and she had a partial response. Now, after I was following with her, I picked her up in about July, taking care of her of this year. And unfortunately, in February of 2019, she started to have symptoms. And on her scan, she had growth of the disease, actually relatively rapidly. So just to briefly explain, we all know about resistance, and mentioning, Ivy and Jill Feldman's work, understanding what resistance each individual patient has is going to be critical at deciding next treatments. So this is from a review from Ali Schramm and Dr. Drillon, looking at what are the potential resistance mechanisms for ROS1. And one of the more common ones is this G2032R. So we have data on this. We know this. Right? We have information that can then help guide patients and you to next trials or next opportunities. So we know that if you develop certain alterations, green means that the medicine works, so crizotinib would work here. But if you develop the G2032R, red, unfortunately crizotinib does not work. And you could see that that resistance mechanism is one of the more tough ones where the drugs do not work well. Cabozantinib has some activity, but this drug does have quite a bit of toxicity or side effects. So we did a Guardant 360 liquid biopsy assay, and we were able to identify this alteration, this ROS1 G2032R, which again might give us further opportunities or options for this patient. Unfortunately, I was not able to get her onto a clinical trial, so there is a drug called TPX005, which is a really good drug because it has activity against this alteration, but that's one of the problems that we see, right, that there are not always slots for the right trials at the right time. So one of the things that I really push my patients and you guys in the room to do is to be aware and be active in understanding sort of what are the trial opportunities in the area, what are the next treatment options available, and discuss those with your doc. It might not be at the institution that you're at, and you may want to partner with another institution or another group because that will give you more options and more opportunities down the line. So interestingly, in this specific patient, because we were not able to get her onto the trial at the time, and because there was some data that lorlatinib might have some activity here, we decided to try lorlatinib. And I won't bore you with the data here, but unfortunately, after 1 month, she progressed on lorlatinib. So the drug, unfortunately, did not have any activity. And that's another thing. It's okay to try drugs, but really push your docs to see whether they're working, and really push your docs to ask, or you tell them, how are you feeling, right? If you're not feeling better, that's a real big red flag, right? It could be toxicity related to the medication, but it also could be the medication not working against your specific subtype of cancer or genetic alteration. So this patient was lucky enough that the month that we put her on lorlatinib, a spot opened up. And we are in the process of getting her onto this study now, TPX005. And I'm fortunate, or I should say hopeful, that she will have a response. But this drug so far in the clinic has been very exciting and very active. So I'm very hopeful that this woman will have a response. And again, very critical here to partner with your different groups and different doctors to find the best opportunities for you. So really, in conclusion, I think this is an exciting time for patients and their care teams. For me, coming to work every day is exciting. You can ask, some folks who I've seen in the clinic. I mean, I couldn't find anything that I'd rather prefer to do on a day-to-day basis. It's a phenomenon. I'm very fortunate to be part of your care. Personal approaches available to select patients, and that's important. Not all patients are going to have a specific alteration, but I think we need to identify those patients. Next generation sequencing, we talked about tissue and plasma is critical in all patients, and we need a biomarker-driven accelerated drug discovery sort of platform and approval platform, and we're starting to see this happen. And really, there needs to be more shared responsibility between the stakeholders, and really that's the patients and the physicians and the industry here. And then again, some opportunities are, participation. If you ever have the ability to participate in a clinical trial that's specifically for you, I think that's a great opportunity for you, but also for folks who come after. And then really understanding the mechanisms of resistance, particularly for the rare alterations where we don't have much data. So, Josh and I were talking about this. For the NTRK fusion, we actually have a drug already available that is in development, right? It's not clinically approved, but the company is already studying a drug that might have better efficacy or activity after lorlatinib unfortunately doesn't work. And then patients and advocacy groups, again, need to drive research and the discovery platform. So I really appreciate your time, and I appreciate you guys listening.